Our next speaker is going to challenge my French. I studied in France, so I speak French. Sebastien Branchu is our next speaker, and he's from AbbVie. AbbVie was known as Abbott previously, and he has continued there. And if we all recall, the Calitra and Norway, one of the first ASTs in the market, HMEs, came through them. So Dr. Branchu, he's gonna talk about leveraging hot melt extrusion to improve bioavailability, which is highly relevant to what we are doing here, and specifically reducing pill burden. Thank you, and I welcome you on the stage. Merci.
Thank you first. Hi, good morning. I'm Sebastien. I work at AbbVie. I live in Germany. I work in the German affiliate of AbbVie. And I want to thank first Ferouz and Simon for organizing and running this very fruitful conference. I want to thank also Ferouz and Simon, as well as my employer, AbbVie, and including my colleagues here, Jeff and Anya, for the opportunity I'm given to present today. And AbbVie has had a long experience of researching and developing and manufacturing enabling formulations, including solid dispersions, which is one type of enabling formulations, to address potential issues of availability for absorption. And this is what I'm going to present today. And in addition to this, AbbVie has had a contract manufacturing business for many years, which means that AbbVie has been making product for other companies for many years. And some of those products utilize enabling technologies. We have a booth, number 7, and please feel free to visit us if you'd like to learn more about these topics. Okay. So here we go. Yeah, thanks, Ferouz, for the introduction. I've been working in those areas of product development and process development for a number of years. I've been at AbbVie for 20 years now. And as has been outlined in some of the previous presentation, there's been a significant risk of poor availability for absorption for a large portion of new orally active candidate drugs. And this is an issue because this can make development times much longer. This can result in an increased pill burden for the patient. This can pose challenges during preclinical and clinical evaluations, and result also in a high cost of goods. So what I'm going to present here is a case study, again, on a product that AbbVie reformulated for a client. And the approach we've used is really the approach we used for any of our products. We have perhaps a dozen marketed products that utilize enabling formulations, including solid dispersions. And this is a typical approach that we use for our development programs. So a pharma company who is an AbbVie client developed originally an oral drug candidate with that was poorly soluble and poorly permeable, and which had a high dose. And that company launched a first generation formulation with a high pill burden, short shelf life, and requiring storage under refrigeration. In parallel, that company contacted AbbVie Contract Manufacturing to develop a second generation formulation to address those limitations. As has been also mentioned previously, biopharmaceutical classification frameworks have been built and used to assess developability in the early stages of research and development. So enabling formulations, as I mentioned, include solid dispersions. And one technique to make solid dispersions is extrusion, hot melt extrusion. And AbbVie has had extensive experience with that technology, including developing, scaling up, validating, and manufacturing commercial products using that technology. In that particular case study that I'm presenting, a stage-based approach was adopted to determine the applicability of the extrusion technology to the particular compound. And with those stages, the compound owner had an opportunity to stop the development program at any time point if the technology proved not suitable for that particular compound. So the 4 stages are formulation development, stage 1, stage 2, process development, stage 3, process scale-up, and stage 4, commercial manufacturing. In stage 1, formulation development, data was generated on the particular selected drug substance solid form to understand the viability of various development scenarios for that drug substance. And that material showed a very poor solubility across a range of media, high dose to solubility ratios, and high crystal lattice energy, which all contributed to a high risk of any successful traditional formulation. By traditional or conventional formulation, I mean a non-enabling formulation. One key acceptance criterion was the ability of the drug to dissolve into at least one excipient matrix under extrusion conditions. And the prototype formulations were initially assessed in the lab using various characterization methods, including lab-scale processing work such as small-scale extrusion. Mixtures were prepared in the lab of drug substances and various excipients to understand mixture behavior, miscibility, solubility, residual crystallinity, and this was done with both binary and multi-component mixtures. What I'm also providing in that presentation, and you can get the presentation afterwards as well, is a whole series of publications, and here you have, the full list of publications with the full description of the references is provided on one of the last slides, and all of those publications, or most of them, are authored by AbbVie and academic partners. And I remember yesterday, someone mentioned Purdue University and Lynn Taylor's group. So Lynn Taylor and her group have been one of the long-term scientific partners of AbbVie, including in particular in that area of solid dispersions. So all I'm presenting here is further detailed in all of those references that I'm mentioning in the slides. Those prototype formulations were evaluated for dissolution and bioavailability. So on dissolution, studies were done to understand drug release mechanisms, and advanced tools were used around imaging, confocal microscopy, nanodroplet measurements, and molecular dynamic simulation. On bioavailability, the second generation formulation that was being developed was found to have bioavailabilities that were more than 200% that of the first generation formulation. And we know it's typical for solid dispersions to provide enhanced bioavailability, but in that particular case, we had a 5-fold improvement relative to a traditional formulation. Stability testing was done on those prototype formulations in relation to various key quality attributes, and in particular in relation to drug release and residual crystallinity. Studies have benefited from knowledge developed, again, that are described further in those publications, and in particular on topics such as polymers' effects on nucleation and crystal growth in solid dispersions, advanced imaging methods around phase separation, and long-term kinetic stabilization of solid dispersions. So the outcome of stage 1 was candidate formulations based on the hot melt extrusion technology that had a high drug load, actually a drug load more than twice that of the first generation formulation, which means that the total dose could be an improved bioavailability, which means the total dose could be reduced by more than 20%, and the drug and the pill burden could be reduced by more than 70%, which is of great importance to patient compliance. And those prototype formulations exhibited acceptable drug release as well as long-term stability at ambient conditions. Stage 2 was around process development. So initially, lab scale work was done on processing using, for example, 18 millimeter scale extrusion. Then the first scale-up was conducted to the 27 millimeter scale to understand equipment configuration aspects such as screw configuration, barrel configuration, again, and the implications of those things on quality attributes such as drug release and residual crystallinity, as well as manufacturability aspects already, right? So at pilot scale, you can already look into very much manufacturability aspects, including the stability of the process. And trials were run in tandem with simulation work, and I'll say a bit more afterwards about simulation. So developing a robust process at pilot scale benefited from work around, in particular, around 4 key process variables that have, some of them obviously have been mentioned in previous presentations yesterday or today. So melt temperature profiles, very important. Spectrometrically measured residence time distributions, torque sensor measured torques, and infrared thermal imaging of the melt. And these are very important variables when developing any extrusion process. And operating windows were developed that provided suitable quality and suitable manufacturability. So a few words here on simulation. Simulation generally can help gain access to non-measurable characteristics, design better experiments, and reduce the amount of material, including drug substance material, needed for development. So I'm providing one literature reference here, also authored by AbbVie, and there's another one. Also, there are at least two references here in this presentation specifically on extrusion simulation work done at AbbVie, and I'm providing here on this slide one example of melt temperature profile as a function of extruder axis under 2 sets of conditions. One condition is a lower viscosity formulation with a lower screw speed, and the other condition is a higher viscosity formulation at higher screw speed. And obviously, all of this simulation work helps guide the experimental studies. The outcome of stage 2 was a robust process at intermediate scale across broad parameter ranges, a rich body of process data, and the ability to go to stage 3. At stage 3, a second scale-up was performed from the 27 millimeter scale to the 40 millimeter scale, first on non-GMP equipment, and the scale-up was based on matching the specific energy input. This was mentioned also yesterday, matching the specific energy input across the scales. And how do we do that? We match melt temperature profiles and residence time distributions. And the move from the 27 millimeter scale to the 40 millimeter scale meant a 3.3-fold increase in throughput. And if you combine this with the longer processing times, you end up with a 10-fold increase in the batch size. Downstream processing work was done to convert the extrudate into a final dosage form that included development of post-extrusion processing steps as well as the use of excipients. And there are also here some good examples from the literature on this type of work. And clinical manufacturing experience helped gather additional data and therefore build additional confidence in the robustness of the process. So the outcome of stage 3 was a robust process at intended clinical and commercial scale, and the ability to go to stage 4. In stage 4, the process was transferred to GMP manufacturing equipment, so 40 millimeter scale extruders, GMP equipment both at the AbbVie facilities and at the client facilities. A validation strategy was developed and a validation protocol was put in place, and 2 batch sizes were validated. The execution of the validation showed the control strategy to be suitable and effective, and the uniform quality to be maintained. And following that PPQ stage, stage 3A of the validation cycle and then stage 3B of that validation cycle helped gather additional data and again build further confidence in the performance of the process. Over the years, process improvement projects have taken place around, for example, cycle time reduction, qualification of alternate sites for drug product manufacturing, qualification of alternate sites for drug substance manufacturing. And all of this obviously has contributed to the successful development program. So in summary, a close collaboration was established between AbbVie and the compound owner that led to a successful long-term program covering both development and commercial stages. AbbVie contract manufacturing successfully applied a hot melt extrusion approach to reformulate an existing product into an amorphous solid dispersion. And the new formulation was validated and brought remarkable benefits to the patient. So the summary of the benefits: pill burden was reduced by more than 70%, the dose was reduced by more than 20%, shelf life was doubled, and food effect was removed. And again, here is the full list of references based on which the slide deck is built. And I want to thank my colleagues at AbbVie. And please feel free to speak to me or write to me with any questions and comments. And again, we have a booth here, booth number 7. Feel free to come to us. Thank you.
Any questions? Yes.
Hi, interesting presentation. I'm Irfan Sayed from Evonik. I've got 2 questions. One is more so on bioavailability enhancement to 200%. Was it because of change in drug loading, or was it because of the process itself, or change in the formulation composition that gave you higher bioavailability?
Yeah.
Sorry, go ahead.
Yeah, sorry. I would say it's the formulation. The physical state of the drug substance in the new formulation is favorable to that, explains that. So we have this stabilized amorphous state, right? Whereas the first generation formulation was a much simpler approach. And so the formulation, I think, explains that improved bioavailability.
Okay. And more so, second question is a general one. We've heard some of the presentations yesterday utilizing lipids for HME solubility enhancement. Have you had any experience developing HME-based process from development to scale up and commercialization using lipids?
Yeah, not in this particular case I've presented, but yes, I believe we have, actually I know we've used lipids at least in development stages on on development stage formulations and products, yes.
Okay. Thank you.
Side of the room. Wakes you up.
Hi, thank you for the wonderful presentation. Just a quick question. Do you think HPM-CAS is suitable for HME? Because we frequently know HPM-CAS will suffer from the thermal degradation, and you will generate succinic acid, this type of acid that can cause some further degradation of your molecule. How often is HPM-CAS used in HME?
Could you repeat your question, because I got only part of it, so I'm not,
Okay.
please.
Yes. How often is
CAS degrades and creates succinic acid that may impact the product stability?
Yes, yes.
Yeah, I'm not too sure about this. We have, in this case, we didn't use that polymer. There are cases where we explored that polymer. I'm not necessarily familiar with all the studies where we use that polymer, so I'm sorry, I don't really have an answer to you, or maybe I would have to look into it further.
Okay, okay. Yeah.
But thank you for the question. Yeah. Yeah.
I can send you a publication. There is a literature where they have exposed HPM-CAS. If you want to send me an email or whatever, I can send you the publication. There is detailed studies on this polymer and what temperatures cause succinic acid to be created. It's going to be really much higher than the usual temperature that it's happening.
Okay, thank you very much, Sebastien.
Thank you.
Thank you.
Thank you.
Good. Thank you.